Pheno Tracker is a deep phenotyping platform that helps generate continuous high-throughput data to characterize disease and intervention. In the next few minutes, I will share with you what the platform does, why it was developed, where we are in its development, and what it can achieve for drug testing and repurposing. Disease in humans is well characterized, mainly because doctors are continuously monitoring patients. Such data allows us to see variability in disease stages, levels of severity, understand how these influence outcomes, and study treatment responses. Such detailed clinical information has led to a paradigm shift in medical practice and clinical trials, stratification of patients, personalized medical treatment, and precision molecular diagnostics that accompany treatment selection. Animal models, on the other hand, which are critical to advance compounds into clinical use, often do not reproduce accurately such detail. For example, animal models and experiment factors are typically selected for lethal endpoints rather than examining natural variability in stages of disease progression that are observed in humans. Natural manifestation of disease in small animals is not well defined, and many questions that would be important for treatment go on unexplored, leading to coarse observations. At the same time, close to 40% of candidate compounds fail to become drugs, in part due to the bottleneck of preclinical development. During this stage, researchers study compounds that show efficacy in cell lines or organoids to understand efficacy and toxicity at the biological system level, which has complex metabolism, immune responses, and tolerance for both disease and treatment. Interestingly, over 60% of animal models used for such testing are mice, rats, or other animals such as hamsters or ferrets. To address the challenge of detailed disease monitoring and quantification of drug responses in preclinical animal testing, we developed a platform for deep phenotyping and deep learning. It is an alternative to an all-present doctor that sees each animal individually 24/7. Importantly, such high-throughput data acquisition from animal experiments often fails due to instrument complexity and high costs, resulting in short-term observation studies with limited sensitivity to account for long-term trends in animal condition physiology without significant invasiveness. That's why the system that we built is a balance between ease of use, cost effectiveness, scalability, and long-term monitoring capabilities. Pheno Tracker is a tracking and analysis platform that is designed to collect continuous data from multiple sensors and extract characteristics of animal physiology, body vitals, behavior, and even the underlying state the animal is in. Powered by deep learning, the system provides real-time alerts, extracts meaningful trends in data, and brings the lab experiment within reach for researchers that are not present or not able to be in the lab when the animal is showing important changes associated with the studied condition. That's why the way the system works is a combination of hardware optimization and software designed to reveal important information that is not obvious from short-term observation. Real-time raw data using thermal and infrared sensors are captured and sent to the cloud for processing. There, each animal is analyzed for posture changes, speed of movement, and other features, including advanced seasonality decomposition and deep learning. Here on the left, you can see the captured information after body detection, and on the right, we show how each 10-minute interval of video is analyzed to reveal patterns using multiple data points. Ultimately, these vary significantly between pre-infection and post-infection conditions, something that might be easily missed if you were to look at the cage for just a few minutes. Once the data is acquired from a sensor, it is separated into individual features, normalized, and prepared for comparison from user-generated metadata. To capture changes in amplitude and periodicity, we separate the trends into individual segments, which are compared to the baseline of the individual animal during the healthy stage before the start of the experiment. Then, data is analyzed for variation from baseline and used to train a predictive machine learning model that incorporates system dynamics into its prediction of outcomes. On top of this process, we developed a software layer to provide real-time view of raw video stream, process data, as well as custom alerts for anomalies and state changes to help incorporate such insights into experiment design and intervention. Additional tools for big data analysis allow the user to compare and contrast the data from a given experiment to accumulated datasets in our proprietary database of animal phenotypic patterns. The interface helps bring the experiment to the user, making it possible to view, explore, and run big data analysis interactively in the cloud, as well as explore the results of this analysis in comparison to ongoing experiments that are being conducted. As a result, this proprietary high-throughput data can be used to train a predictive model of animal disease progression that allows a detailed analysis of phenotypic and molecular data to extract biomarkers associated with pathogen type, stage of disease progression, treatment response, recovery rate, and even population dynamics of animal-to-animal interaction or disease spread, continuously monitoring these conditions and changes. For each stage, animal and organ molecular profiles using RNA-seq can show how the body reacts to infection, what types of pathways are activated, and how this can be used to describe the biology of infection that drives disease progression. Our team has been working on several different prototypes, viruses, vaccines, and treatments working with labs at University of California, San Francisco, Tulane University, and Louisiana State University. We tested and refined the hardware for use in Biosafety Level 2 and Biosafety Level 3 facilities, developed solutions for different animal models, designed and tested our software with lab technicians and research users, and as a result, we have collected data on enteroviruses, COVID-19, compared routes of infection, and now we are testing interventions for efficacy and toxicity. In the next 6 months, we plan to A. Complete ongoing experiments and publish findings with our research collaborators. B. Develop several more cages based on demand from partner labs and test new sensors for specialized experiments. C. Ultimately, we plan to demonstrate how this type of data can be utilized for basic research, drug testing, as well as characterization of treatment response to optimize and shorten preclinical testing. In reality, there are so many unknowns about disease in animal models, and if we are to use animals responsibly and understand how these biological systems can mimic human disease and help us develop novel interventions, we have to develop a model of personalized disease dynamics. High-throughput data and precise understanding of how disease develops will ultimately open the door for new treatments, new knowledge, new capabilities. Our goal is to make this platform a standard part of all animal experiments and a major component of animal testing. Together with our collaborators, we are working on a number of high-impact projects that include SARS-CoV-2 treatment testing, a variety of vaccines, as well as experiments that can help optimize intervention timing and prevent transmission. We believe that this technology is a powerful platform for pharmaceutical model of drug discovery, testing, and drug repurposing. With so many companies turning to AI-guided drug design, analytical drug repurposing models, and discovery of novel compounds, Pheno Tracker is a competitive advantage in the hands of experts with insights into animal condition, testing strategies, collected baseline data, as well as the capability to help design novel sensors that provide unique insights into the many aspects of health and disease. Thank you for taking the time to learn about Pheno Tracker. The studies I described today were done in collaboration with UCSF, Tulane, and LSU, and were funded by DARPA, NIH, and NSF grants. For any additional questions, feel free to reach out to me, Ilya at pine.bio, or visit our website, pheno.tracker.com.